In this video, the wave brain concept, which has gained recent attention, suggests that consciousness is linked to endogenous electromagnetic fields generated by neuronal activity. Studies, such as Galinsky's, demonstrate the presence of waves in the cortex that play a key role in these fields, with corticothalamic pathways and deep brain neurons, particularly in the thalamus, being central to this process. Building on this, an inhomogeneous anisotropic model of the brain cortex predicts the emergence of weakly damped wave-like modes in the cortical layers. These modes, which challenge traditional views of signal propagation, have been confirmed through numerical simulations, showing prolonged activity that doesn't correlate with neural fiber directions. This aligns with experimental studies revealing electrostatic field activity in the form of traveling waves, sometimes observed even without direct neuronal connections. Furthermore, nonlinear coupling between multiple wave modes can produce both high-frequency spiking activity and low-frequency rhythms, offering new insights into the dynamics of brain waves. Electromagnetic field theories suggest that consciousness arises from dynamic electromagnetic fields generated by neuronal currents. To conclude, it's important to highlight a compelling theory regarding the origins of the brain's endogenous electromagnetic field and its potential role in consciousness, as explored by Hales. This work addresses the long-standing challenge of linking EM phenomena to consciousness by proposing a biophysically realistic mechanism. It suggests that the coherent action of transmembrane ion channel currents, which generate electric and magnetic fields, may be the primary source of the brain's EM field. These ion channels function like tiny current filaments, creating structured, focused fields that can influence nearby neurons, akin to sweeping lighthouse beam. This mechanism not only provides insight into the origins of EEG and MEG signals, but also suggests a new way to understand the subjective nature of perception. By positing a decomposition of the EM field that reveals both objective and subjective perspectives, Hales offers a physically plausible explanation for how these fields might give rise to the experience of consciousness, addressing some aspects of the hard problem of consciousness. The idea of electromagnetic consciousness involves understanding how the interactions of electromagnetic, EM, fields at various biological scales contribute to the phenomena of consciousness. This concept can be broken down into two levels, from atom to neuron, focused on biophoton release within mitochondria, and from neuron to brain, concentrated on the action potentials resulting from ionic movement through neural structures, such as the sodium-potassium pump. Each level generates electromagnetic fields of different frequencies, affecting how information is processed and integrated within the nervous system. Below is a detailed exploration of each level. First, we look at from atom to neuron, based on biophoton release in mitochondria. At the microscopic level, biophotons emitted from mitochondrial processes in neurons generate high-frequency EM fields. These photons might influence intercellular signaling, quantum coherence, and potentially the synchronization of individual neurons, contributing to the fundamental building blocks of consciousness. Biophotons are ultra-weak light particles emitted by biological cells during various metabolic processes. In neurons, biophoton production primarily occurs in the mitochondria due to oxidative metabolism and reactive oxygen-species interactions. Mitochondria emit biophotons in the ultraviolet and visible light spectrum, corresponding to high-frequency electromagnetic fields, ranging from approximately 10 to the power of 14 to 10 to the power of 15 Hz. The emission of biophotons at the atomic and molecular level within mitochondria represents quantum interactions, where molecules transition between different energy states, releasing photons. The high-frequency biophotons could play a role in cellular communication and quantum coherence within the neuron. The idea is that these photons may enable neurons to achieve synchronized states, contributing to the underlying mechanisms of consciousness at the cellular level. Biophotons might facilitate intercellular signaling, potentially affecting gene expression, metabolic regulation, and even neuronal excitability. Within the neuron, biophotons can interact with various intracellular components, including microtubules, which have been proposed to play a role in quantum consciousness theories, e.g., the orchestra-OR theory. This interaction might allow for the maintenance of quantum coherence within the neuron, providing a possible link between cellular-level processes and conscious awareness. At the macroscopic level, neuronal networks utilize low-frequency electromagnetic fields generated by ionic movements, via the sodium-potassium pump and action potentials. These fields facilitate the synchronization and integration of neural activity across the brain, producing the global, unified field necessary for conscious awareness. At the neuronal level, consciousness is linked to the propagation of action potentials, electrical signals generated by the movement of ions, primarily sodium and potassium, across the neuron's membrane. The sodium-potassium pump plays a crucial role in maintaining the ionic gradients that drive these action potentials. The sodium-potassium pump and action potentials generate electromagnetic fields in the low-frequency range, from approximately 1 Hz up to 1 kHz. This frequency range corresponds to various neural oscillations, brain waves, such as delta, 0.5 to 4 Hz, theta, 4 to 8 Hz, alpha, 8 to 13 Hz, beta, 13 to 30 Hz, and gamma, 30 to 100 Hz. The EM fields generated by neuronal action potentials can interact and influence each other, resulting in synchronized neural oscillations across different regions of the brain. This synchronization is believed to be essential for the integration of information and the formation of conscious experiences. Neural networks communicate through the combined action of these low-frequency EM fields, enabling large-scale brain activities. The collective synchronization of neurons via these fields is thought to underlie the global binding of sensory inputs, thoughts, and memories into a unified conscious experience. Different frequencies of neural oscillations are associated with various cognitive and perceptual states. For example, gamma oscillations, 30 to 100 Hz, are linked to higher-order processing and conscious awareness, while delta and theta waves are associated with sleep and subconscious processing. The sodium-potassium pump indirectly contributes to this process by establishing the resting membrane potential, which is necessary for the generation and propagation of action potentials and, consequently, the creation of low-frequency EM fields. In this slide, we explore the detailed mechanisms of ion channels and their relationship to electromagnetic field equations, illustrating how the brain's endogenous EM field originates from neural activity. This examination delves into the biophysical processes that allow EM fields to scale from individual neurons to complex, brain-wide interactions. Let's start by discussing ion channels. Background transporters establish the cell's resting potential by creating par-membrane charge density profiles, which counteract ion loss from active transport through ion channels. Rapid, coherent ion traffic generates distinct electric and magnetic fields that stand out over slower background processes. We include an image that shows the dynamic electric field, E, R, T, and magnetic field, B, R, T, characteristic of a transmembrane current filament. It illustrates how rapid ion movements create localized charge disparities in the membrane regions, which extend into both extracellular and intracellular spaces. The magnetic field strength directly follows the transmembrane current's time course, highlighting how the rapid ion traffic is central to generating these fields. Additionally, we present the lumped element method for modeling ion channels, a key approach in neural signaling. This method treats ion channels as part of an interconnected electrical system, helping us understand their role in generating the brain's larger-scale EM phenomena. On the right, we compare Maxwell's macroscopic equations with their microscopic counterparts. This comparison shows how these equations can be adapted to brain tissue to describe the origins of EM fields. Brain tissue acts as a quasi-static convector, producing fields with negligible radiation, as changes in magnetic fields are too slow to contribute to radiative effects. We conclude by highlighting key points. EEG and MEG signals arise from the quasi-static fields produced by neurons. Electromagnetic coupling allows the field system of one neuron to affect neighboring neurons, even without direct contact. Magnetic field activity in neural tissue is driven by current density, not just current. This synthesis provides an overview of how ion channel activity contributes to the brain's endogenous EM field, connecting microscopic processes to measurable macroscopic phenomena. This slide illustrates the coherence and dynamics of local electromagnetic (EM) fields in single neurons and provides a multi-scale perspective on their sources and interactions. On the left side, we explore the different parts of EM field sources within individual neurons, broken down into three distinct levels: microscopic, mesoscopic, and macroscopic. At the microscopic level, NM, the diagram A zooms in on the ion channels embedded in the neuronal membrane. These ion channels are responsible for generating localized electrostatic fields through the movement of ions across the cell membrane. As ions move in and out, they create charge differences that produce static electric fields around the channels within the intracellular (ICS) and extracellular spaces (ECS). At this scale, magnetic effects are minimal, and the fields primarily result from the electrostatic interactions near the ion channels. This localized activity sets the foundation for the neuron's overall EM field. Shifting to the mesoscopic level, the diagram B expands the view to encompass the entire neuron, including the soma, axon, and dendrites. Here, the focus is on the coordinated activity of multiple ion channels distributed throughout the neuron's structure. As these ion channels open and close, they generate fast dynamic electric fields and magnetic fields, creating quasi-convection EM fields. This bipolar current flow, influenced by the neuron's geometry and the synapse locations, gives rise to more complex field patterns that fluctuate over time. These interactions at the mesoscopic level show how local field dynamics within a single neuron contribute to its role in neural networks. Moving to the macroscopic level, CM, in diagram C, we see how these EM fields scale up to influence larger neural networks and brain regions. At this level, the neuron participates in generating quasi-electrostatic fields characterized by slower dynamic electric changes. These fields result from large-scale polarization across the ECS and ICS, extending through various cortical layers and even influencing surrounding tissues, including the dura and skull. The macroscopic fields illustrate how local neuron activity contributes to the brain's overall electromagnetic environment, interacting across different scales. The right side of the slide presents the total EM field of a single neuron, highlighting its dynamic nature. The top diagram provides a structural overview of the neuron, including the soma, axon, and branching dendrites. This structure indicates how EM waves propagate directionally through these different regions. Below, the time-sequenced images display the evolution of the EM field over time, showing how the field changes in strength and direction in response to ion channel activities. These visualizations, marked with arrows and color gradients, depict areas of varying field intensities, illustrating the neuron's dynamic electrical activity. This coherent activity acts like lighthouse beams, sweeping across the surrounding neural structures and contributing to the brain's dynamic electromagnetic landscape. In summary, this slide demonstrates that the coherent generation of EM fields at the microscopic, mesoscopic, and macroscopic levels creates a dynamic interplay essential for neural communication. The integration of local EM field sources within individual neurons scales up to influence the brain's global EM environment, potentially playing a key role in the processing and integration of information. Understanding these multi-level interactions provides insights into how the brain's electromagnetic fields might contribute to more complex functions, possibly extending to consciousness. The electromagnetic (EM) fields produced by individual neurons do not remain isolated. When groups of neurons fire synchronously, their fields combine to form coherent electromagnetic waves. This coherence is crucial for effective neural communication, allowing EM waves to propagate throughout the brain. These waves interact over larger distances, creating the brain's dynamic electromagnetic environment. Acting like sweeping lighthouse beams, these waves focus and direct information across neural networks, potentially serving as a mechanism for coordinating complex brain functions. This slide proposes that consciousness might arise from the structured dynamics of the brain's EM field. The brain's EM field is not merely a byproduct of neural activity, it could serve as a medium for integrating information across various regions. This aligns with the electromagnetic correlates of consciousness theory, which suggests that the unified and coherent nature of the EM fields within the brain corresponds to the subjective experience of consciousness. On the left side, we explore the origins of these EM fields within the brain's cortical gray matter. Figure provides a cross-sectional view of the nervous tissue, illustrating the dense network of neurons and astrocytes. Each of these cells forms an intricate, branched structure, partitioning the tissue into intracellular, ICS, and extracellular spaces, ECS. The ECS, depicted in red, is a narrow sheet/tunnel matrix ranging between 40 to 110 nanometers in width. Despite its narrowness, the ECS has a high tortuosity, occupying approximately 10 to 20% of the brain's volume. Ion channel activity across cell membranes, highlighted in Figure B, generates both electric and magnetic fields. The line AA represents the distribution of static charge, electric field strength, and potential at different membrane intersections. The arrows indicate the direction of the resting electric fields, while blue and black arrows show the transmembrane currents in the ECS and ICS. Figure D presents the foam-like structure of the nervous tissue, illustrating how these tortuous pathways lead to the decoherence of current flow at the microscale. This dynamic environment, resulting from the interaction between intracellular and extracellular spaces, forms the basis for the brain's endogenous EM fields. Shifting to the right side, we focus on the functional implications of these EM fields in neural processing. The figures compare two conceptual models to understand the role of EM fields in the brain's signaling system. A represents the traditional AP closed-loop, EM open-loop model, where EM fields are viewed as epiphenomenal, mere byproducts of neural activity without any causal influence. In this model, neural communication is primarily driven by action potentials, which are strong but slow and do not rely on line of sight. EM fields are fast and weak but do not contribute to feedback within the neural circuits. In contrast, Figure B presents the AP/EM closed-loop model, suggesting a more dynamic role for EM fields. Here, both action potentials and EM fields form a closed-loop interaction, allowing for feedback mechanisms that can influence neural activity. The interplay between slow, strong APs and fast, weak EM signals creates a dynamic neural network. Recent empirical evidence supports this model, indicating that EM fields might actively participate in neural communication rather than being mere reflections of neuronal activity. This involvement of EM fields in a feedback loop may be fundamental to the brain's information processing capabilities, potentially contributing to the emergence of consciousness. Together, these images capture the journey from neural action potential to consciousness. The left side explains the physical origins of EM fields in the complex neural structures, highlighting how coherence among EM fields forms a dynamic network capable of integrating information across different regions of the brain. The right side proposes that the EM field's functional role involves more than simple signaling; they act as a medium for coordination and feedback within neural circuits. The coherence of EM fields, resulting from synchronous neuronal firing, is akin to sweeping lighthouse beams that guide information across the brain. This suggests that consciousness might emerge from the structured dynamics of these EM fields. By embracing this view, we align with the electromagnetic correlates of consciousness theory, which posits that the unified and coherent nature of EM fields could correspond to the subjective experience of consciousness. Thus, this slide underscores the idea that EM fields are not just a byproduct of neural activity, but are integral to the brain's dynamic processes that give rise to conscious awareness. To dig deeper into the electromagnetic neural field behaviors in the following slide, we are going to present the universal theory of brain waves, which is introduced by Vitaly L. Galinsky. This slide provides a deeper understanding of how neural electromagnetic fields transition from linear oscillations to nonlinear synchronized spiking and collective brain rhythms. The theory focuses on modeling the brain's complex electromagnetic interactions, emphasizing both their dynamic behavior and the impact of the brain's inhomogeneous, anisotropic medium. The left side of the slide presents a mathematical framework for understanding how collective synchronous spiking in neural networks can be modeled as coupled nonlinear oscillators. Here, the first set of equations encapsulates the nonlinear interactions of wave modes using a Hamiltonian approach. The formulas describe how brain wave modes are influenced by both harmonic, basic oscillatory, and anharmonic, nonlinear, components. The harmonic term accounts for the basic oscillatory behavior, while the anharmonic term explains interactions between different wave modes, highlighting how coupling between modes results in nonlinear, complex spiking activity. Additionally, the wave modes are represented through temporal and spatial amplitude expressions, linking them to the overall wave field in the brain. The Fourier integrals describe how these modes evolve over time and space, which contributes to the dynamic landscape of brain waves. This mathematical framework provides a bridge between simple oscillator models and more intricate neuron models that involve pulse-coupled spiking. The plot at the bottom left visualizes how linear and nonlinear oscillations manifest within neural networks. The spikes shown represent the transition from regular oscillatory behavior to more complex, nonlinear dynamics, which occur at different levels of neural excitation. This visualization emphasizes how nonlinear interactions are crucial for generating the synchronized patterns seen in brainwave activity. The right side dives into the brain's electromagnetic activity, describing it through Maxwell's equations adapted for an inhomogeneous and anisotropic medium. These equations capture the interactions between electric fields, magnetic fields, current flows, and charge distribution within brain tissue. They illustrate how the brain's complex, varying conductivity and permittivity affect the propagation of electromagnetic waves. One of the equations focuses on the spatial-temporal evolution of the electric potential, accounting for the anisotropic nature of brain tissue. This formulation shows how electric fields generated by neural activity are influenced by the brain's medium, contributing to the formation of various wave modes. Additionally, the use of an anisotropic conductivity tensor reveals how electrical signals propagate differently depending on the direction within the brain's fiber structures. The visualizations at the bottom right depict wet cow modes, wave energy transfer coherent oscillatory wave modes, distributed randomly within the brain tissue. These snapshots highlight how brain waves are not uniform but instead show localized concentrations of wave activity. The colors represent a spectrum of frequencies, ranging from low, blue, to high, red, indicating the brain's complex, multi-frequency electromagnetic environment. These images underscore how waves propagate through the brain, constantly changing in response to internal and external stimuli. Together, the equations and visualizations on this slide provide a comprehensive model for brain waves, extending from simple linear oscillations to complex nonlinear dynamics. Galinsky's theory moves beyond basic oscillator models, incorporating nonlinear interactions that result in collective synchronization across neural networks. The Maxwell-based framework further explains how these wave patterns are shaped by the brain's unique structural properties. This universal approach to modeling brain waves offers insights into how synchronized neural activity and electromagnetic fields may work together, ultimately contributing to brain functions such as signal integration and information processing. By capturing both the nonlinear dynamics of neural oscillators and the electrodynamics of brain tissue, the theory lays the groundwork for exploring the emergence of complex brain rhythms and potentially the underpinnings of consciousness. This slide provides a comprehensive look at the complete wave mode trajectory through cortical tissue, examining how brain waves behave as they propagate through the complex environment of neural networks. The visualizations and graphs presented illustrate how the spatial and temporal characteristics of brain wave modes are influenced by critical parameters, thereby affecting neural activity. The top left portion of the slide depicts the analytical probability density spectra of brain waves as a function of the spatial criticality parameter S/SC. This criticality parameter reflects the conditions under which brain waves transition from linear to nonlinear behaviors. The curves in the graph show how the probability density changes with varying values of S/SC. As the system approaches different states, indicated by changes in the critical parameter, the distribution of wave mode amplitudes exhibits a distinct scaling pattern. This analysis is crucial because it provides insight into the conditions that lead to nonlinear dynamics, such as spiking activity within neural networks. Adjacent to this, the middle graph illustrates the size distribution of wave modes in relation to the number of electrodes. This plot indicates how the likelihood of encountering larger wave modes decreases as their size increases, revealing a particular scaling behavior. Understanding this distribution is significant because it reflects the collective neural activities and how they propagate through the brain's tissue. The mode sizes and their interactions contribute to the formation of the brain's overall electromagnetic environment. The bottom left plot delves further into the criticality analysis by multiplying the probability density spectra with a scaling factor. This reveals how the distribution behaves across different values of the phase shift phi. The impact of this phase shift on the spatial criticality parameter indicates how neural oscillations transition to more complex dynamics, influencing the emergence of nonlinear behaviors like spiking. This plot emphasizes the importance of phase relationships in the overall dynamics of brain waves. On the top right, the slide presents spatial and temporal probability density spectra obtained from an ensemble of 1 million wet cow modes. These modes are randomly distributed and propagated through cortical tissue, and their probability densities are visualized using a smooth gradient of colors. This representation captures how oscillatory signals evolve across space and time within the brain, reflecting changes in wave mode criticality. The varying colors indicate how the wave mode's probability distributions shift, illustrating the intricate nature of neural signal propagation. The brain visualizations on the right side of the slide offer a more tangible depiction of how these wave modes manifest as trajectories within the cortical tissue. The center images show temporal signals, depicted as spikes, which represent different values of neural activity. These snapshots provide a glimpse into the complex wave propagation pathways in the brain, highlighting how wave modes travel through neural structures over time and contribute to the brain's dynamic electromagnetic environment. At the bottom right, two examples of complete wave mode trajectories are displayed. These images show the paths of wave modes under randomly selected parameters and initial conditions, illustrating the variability and complexity of wave propagation in the brain. The colored lines trace the trajectory of wave modes as they move through different cortical regions, reflecting how initial conditions and parameters influence the spatial and temporal dynamics of brain waves. This visualization underscores the diverse nature of neural oscillations and their potential to form localized regions of activity. The underlying theory for this slide involves equations describing nonlinear oscillatory amplitudes, which govern the evolution of wave modes' amplitudes and phases. These equations, referenced in the slide, incorporate terms that account for the criticality and nonlinear interactions of wave modes. As the criticality parameter S/SC changes, the system transitions from linear oscillations to nonlinear regimes, resulting in complex waveform patterns and spiking activities. Additionally, the spatial distribution of wave modes within the brain plays a pivotal role in shaping these oscillatory dynamics. In summary, this slide provides a detailed exploration of how spatial and temporal dynamics influence the behavior of wave modes in the brain. By analyzing probability density spectra, mode sizes, and wave trajectories, we gain insights into the complex processes underlying neural synchronization and electromagnetic field propagation. This slide presents the results and applications of the concepts discussed in the previous slides, focusing on the electromagnetic wave propagation and their effects on neural dynamics. It showcases both theoretical electromagnetic wave interference and practical examples of wave trajectories within the brain. On the top left, we see an image representing electromagnetic wave interference patterns. This visualization highlights how electromagnetic fields can form complex interference structures when multiple wave sources interact. These patterns are crucial for understanding how EM waves propagate and interact in various environments, similar to how neural waves interact within the brain. To the right, there are two frames from videos demonstrating the propagation of electromagnetic waves within an elliptical medium. These videos visualize how electric fields evolve over time within an enclosed space, illustrating the dynamics of wave propagation in a controlled setting. The top middle and right images represent different states of this propagation, showing how the electric field distributes spatially in response to boundary conditions within the ellipse. This simulation mirrors how EM fields might behave in the brain's curved, complex geometry, where tissue properties and boundaries affect the wave trajectories. The bottom row transitions from theoretical models to practical examples, displaying brain electromagnetic wave trajectories. The leftmost image provides a detailed view of a human brain with colored lines indicating the paths of EM waves as they travel through various neural regions. These trajectories are derived from the wave mode dynamics discussed in previous slides, showing how electromagnetic fields move through the brain's intricate structures. The two images on the bottom right represent frames from videos that further illustrate the temporal evolution of these wave trajectories within the brain. Each video demonstrates how these waves propagate over time, adapting to the brain's morphology. The varying paths reveal the influence of brain tissue properties, anisotropy, and inhomogeneity on wave movement. These visualizations are directly connected to the earlier discussions on spatial and temporal probability density spectra and wave mode criticality, offering a concrete example of how electromagnetic fields translate into dynamic neural activities. These results are a direct application of the theoretical framework discussed in earlier slides. The interference patterns at the top reflect the complex interactions of wave modes, like the multi-mode wave interactions occurring in the brain. The wave propagation in the elliptical medium models how electromagnetic fields might move through brain-like structures, accounting for curvature and boundary constraints. The brain wave trajectories at the bottom then illustrate these concepts in a biological context, showing how wave modes navigate the brain's neural networks. In essence, this slide demonstrates the culmination of the theoretical models of electromagnetic wave dynamics and criticality. It offers a visualization of how the brain's electromagnetic environment evolves in real time, contributing to the understanding of how neural synchronization, information integration, and potentially consciousness arises from these underlying wave dynamics. In the earlier slides, we discussed how local electromagnetic fields generated by neuronal activity can propagate, interfere, and form complex patterns across different brain scales. The dynamic interplay between wave modes, both spatially and temporally, contributes to the brain's overall electromagnetic environment. We also saw how these wave patterns could evolve into intricate trajectories, influenced by factors such as criticality, nonlinear interactions, and the brain's anisotropic properties. This slide extends these findings by illustrating how inward-directed electromagnetic fields may serve as a foundation for conscious experience. On the left, we see how synaptic currents, particularly in upper cortical layers, produce charge paths that flow bidirectionally, creating fields above and within the cortex. Crucially, the inward-directed fields concentrate toward the brain's center, generating strong EMFs that potentially interact with deeper brain structures, like the thalamus and brainstem. This aligns with the earlier notion that coherent wave modes within the brain contribute to an integrated electromagnetic environment that supports higher-order neural processes. On the right, we explore how these electromagnetic interactions relate to different states of consciousness. The attractor plots derived from EEG data illustrate how brain wave complexity changes across states: awake, loss of consciousness, anesthesia, and recovery. In the awake state, the chaotic attractor plot exhibits a more spherical shape, indicating a rich and dynamic EM field environment. In contrast, during LOC and anesthesia, the attractor flattens, reflecting a reduction in the brain's EM complexity and thus a possible disruption in the coherence of inward-directed electromagnetic fields. Upon recovery, the attractor resumes its spherical form, suggesting a re-establishment of the integrated EM field that may correspond to the restoration of consciousness. By concluding with this slide, we link the theories and visualizations from the previous slides to the notion that the brain's endogenous electromagnetic waves, particularly those directed inward, are closely tied to the mechanisms of consciousness. The interplay of these wave modes, their coherent integration, and their influence on neural structures present a compelling model for understanding how consciousness could emerge from the brain's electromagnetic activity.